The cystosomes are also known as blood trematodes or blood flukes. Okay. So now the classification of cystosomes, the zoological classification of cystosomes. The cystosomes fall under the kingdom Animalia, the phylum Platyhelminthes, which includes the worms which have flat body. So they fall under the phylum Platyhelminthes, and the class Trematoda. The class Trematoda includes those worms which have a large prominent sucker with a hole in between. So the term Trematoda is derived from the Greek word trema, which means a hole-like appearance. As this worm has a hole in its suckers, so it falls under the class Trematoda. And the order Schistosomatidae, family Schistosomatidae, and the genus Schistosoma, and species there are mainly 5 different species under this genus Schistosomes, Schistosoma, which include the species like Schistosoma hematobium, Schistosoma mansoni, S. japonicum, S. mackonji, and S. intercalatum. So among these these 5 species, the mostly clinically important species are the first 3 listed one, that is Schistosoma hematobium, mansoni, and japonicum. These 3 are mainly clinically important species. And among these different species, we are focusing on the Schistosoma hematobium only. So this is the zoological classification. Now, introduction about the Schistosomes. So the genus Schistosoma is also it was formerly called as Bilharzia, which after its discoverer, that is Theodore Bilharzia. This Theodore Bilharzia first identified this schistosome in the mesenteric vein of an Egyptian in Cairo. So this worm was formerly called as Bilharzia. So now later it was renamed as Schistosoma. So why is it renamed as Schistosoma? We will know in later. So now the talking about the schistosomes. The schistosomes are clinically important worms which cause the disease schistosomiasis. So the disease caused by schistosomes is known as schistosomiasis. So schistosomiasis is a infection caused by schistosomes. Now the schistosomes are dioecious in morphology. That is, they have 2 different sexes. That is, the male and female worms are separate. So schistosomes are dioecious, and they have no any second intermediate host. And the schistosomes live in the venous plexus of definitive host, and the location of these worms varies with the species. So the schistosome Hematobium, Schistosoma mansoni, and Schistosoma japonicum collectively cause a disease known as schistosomiasis or bilharziasis or snail fever. So schistosomiasis is a disease caused by the worm Schistosomes, which includes these three clinically important species. And the Schistosoma meconici and Schistosoma intercalatum are less frequently associated with schistosomiasis. So these three are the most important ones. Now talking about the geographical distribution of schistosomes. Schistosomiasis is estimated to affect almost 200 to 300 million people every year, and up to 200 to 300 million people, and cause 75,000 deaths per year worldwide. So schistosomiasis affects 200 to 300 million people and cause 75,000 deaths per year worldwide. And the schistosomiasis is mainly an important public health problem in the Africa, Asia, and Latin America. So it is mostly common in Africa, mostly then Asia and Latin America. Among these three different species, the Schistosome, Hematobium, and Mansoni are mostly widespread in Africa. The Mansoni, Schistosoma Mansoni is also found in it is found in Africa, and beyond that, it is also found in some parts of South America and West Indies. And the Japonicum, Schistosoma Japonicum is restricted to the Far East, and especially in the Japan. So the Hematobium and Mansoni are mostly found in Africa. Mansoni is also found in South America and West Indies. Japonicum is found mostly in the Far East and most common in Japan. And Schistosoma meconici is restricted to Laos, Cambodia, and Thailand. So it is mostly common in Thailand, Mekong River Basin. And intercalatum is mostly found in the central and western part of the America. So this is the geographical distribution. So here in the map, you can see that the geographical distribution of these different species of Schistosoma. So the Mansoni is found in the South America. Here in the map, the red part is the South America, and Hematobium is found in the Africa. Hematobium and Mansoni both are found in the Africa also. And Mansoni and Hematobium are found in Africa, and the Japonicum, species Japonicum is found in the Far East, that is mostly in Japan, and intercalatum is found in the south, central and western part of Africa, and Mekongji is found in the Thailand. So now general characteristics of schistosomes. So we learned about the geographical distribution of schistosome, its geological classification. Now it's we are going to study about its general characteristics. So the schistosomes have separate sexes, that is, they are dioecious in nature, in their morphology. But the genus, they fall under the genus trematode, and the trematode are usually hermaphrodite in morphology, that is, the male and female sexes are found within the same worm, but this schistosome differ from that because it has separate sexes, that is, the male and female are separate. And the body, the trematode, which includes which falls under the phylum Platyhelminthes, or the which includes the flukes, which have flat body, but the cystosomes differ from that because it doesn't have a flat leaf-like body, but has a cylindrical body resembling with that of roundworms. This might be because that it is within the definitive host. This worm is found within the venous like a vesical plexus of the definitive host. So this might be the this morphological features might be due to its adaptation to the vesical plexus of the definitive host. So in order to stick to the vesical plexus, that is the blood vessels surrounding the urinary bladder, this worm has a cylindrical body resembling the roundworm. So talking about the male and the female sexes, the female has a long body than the male, but the male is broader than the female. And the male, the lateral borders of the male are rolled ventrally to form a gynecophoric canal. So the lateral borders of the male are rolled ventrally so that a canal-like structure is formed, that is called as a gynecophoric canal, in which the female is held. So though the sexes are separate, the male and female worm live in close proximity. So the male worm holds the female worm in its gynecophoric canal. Now, since the male, the body of the male seems like it is split longitudinally into two parts, hence the worm are named as cystosoma, which is derived from Greek word cysto means split and soma means body. Due to this, the male has a lateral borders roll ventrally, making a canal-like structure, hollow canal-like structure. The body appears to be split into longitudinally. So the worm was later named as cystosoma. So the cystosomes have two suckers in their morphology, which are anterior and posterior. They are also called as anterior and posterior sucker, or the anterior sucker is present surrounding the mouth, so it is also called as oral sucker, and the ventral or posterior sucker is found beneath the pharynx. So the worm has a the worm has a ventral sucker which bears spines, small spines, which help the worm to stick to helps the worms to stick to the vesical plexus of the definitive host. So This worm lacks muscular pharynx. All other trematodes bear the pharynx, but the cystosoma lacks this. So it differs from the trematode in these characteristics. And the intestinal ceca of the cystosome reunite after bifurcation to form a single canal. So in other trematodes, the esophagus gets bifurcated into two parts, and it end in the intestinal ceca. The esophagus bifurcates into two intestinal ceca, and they do not meet in other trematodes, but here in the cystosoma they get reunited after the bifurcation to form a single canal. So this the cystosome differ in these characteristics from other trematodes. And the number of testes in the male cystosome varies between 4 to 8. So in the male worm, there are 4 to 8 testes. Uh, the other trematodes bear the operculated eggs, while this cystosome bears the non-operculated egg. So this egg not float in the salt solution. So this cystosome differ from other trematodes in this non-operculated egg. So and also the trematodes, other trematodes bear the radia stage in their life cycle, while this cystosoma does not bear the radia stage. So it is different. It differs from other trematodes in these characteristics also. So the circaria of the trematode of this cystosome is has a forked tail, that is it has so it differs from other trematodes in these characteristics also. So due to the presence of this forked tail, the cystosomes can penetrate the unbroken skin of the definitive host. So this is the general characteristics. Now we are going to talk about the morphology of cystosomes. So the three clinically species of the cystosomes resemble each other very closely. Uh, so it is the species are differentiated on the basis of their morphological characteristics of the adults and the eggs. So these three species resemble each other closely. Uh, the female worm is longer than the male, but the male is broader than the female. So the adult worm has a size of 1 to 2 centimeter, that is the length of 1 to 2 centimeter, and the breadth of 0.25 to 0.5 millimeter, while the egg has a size of 70 to 170 micrometer by 40 to 70 micrometer. And it is oval in its structure and bears spine. So by looking at the location of the spine, the different species of these cystosomes can be differentiated. So the egg contains a fully developed embryo. Within the egg, there is a fully developed embryo, which is called as miracidium. This is a morphology. So here in the chart, you can see the different characteristics of different species of cystosomes. So here the adult of the location of the cystosomes varies in the definitive host. That's it. The adult of the Schistosoma hematobium lies in the veins surrounding the bladder, that is vesical plexus. So the adult worm of the Schistosoma hematobium is found in the vesical plexus. That of Manzoni is found in the inferior mesenteric vein. That of Japonicum is found in the superior mesenteric vein, and that of Meconium and intercalatum are found in other mesenteric veins. So the location of the adult worm within the definitive host varies. So the length of the uh, adult worm also varies within species. So here you can see Schistosoma hematobium has a length of 10 to 15 millimeter by 0.75 millimeter, that is male, and the female has a length of 16 to 20 millimeter by 0.25 millimeter, and so on. You can see in the chart. So the adult worm of these different species has different length and sizes. So the eggs of these different species also vary in size and morphology. So the egg of Schistosoma hematobium has a size of 140 to 170 micrometer by 40 to 70 micrometer. So other species have different sizes. So here in the chart you can see that, and the location of the spine also varies. So the Schistosoma Schistosoma hematobium has a spine which is it has a terminal spine. The Manzoni has a lateral spine, and the Schistosoma hematobium has a terminal spine. Manzoni has a lateral spine, and Japonicum has a lateral spine which is round knob-like in structure. So this is by looking at this different location of the spines, the eggs of the different species can be identified. So the talking about the distribution, geographical distribution, hematobium is found in Africa. Manzoni is also found in Africa and other parts of South America. Japonicum is towards the Far East, mostly in Japan. Megonji in Thailand. Intercalatum in Central and Western Africa. So these are the different geographical distribution, and the reservoir host of the species are also mostly the humans are the reservoir host of different species, except the Japonicum, which has other animal host as its reservoir. So intermediate host of these different species also varies. That is, the hematobium, the intermediate host of these different species of cystosomes is the snail, and the different genus of the snails are its intermediate host. That is, the Schistosoma hematobium has an intermediate snail host of the genus Bulinus, and that of Mansoni is the genus of the genus Biofalaria, and so on. These, by looking at this chart, you can get a rough idea about the cystosomes, its morphological and geographical distribution, and different its intermediate host, and so on. So now we will talk about Schistosoma hematobium. So the worm Schistosoma hematobium was formerly called as Bilharzia hematobium after its discoverer Theodore Bilharzia. Theodore Bilharzia identified the worm, so it was formerly called as Bilharzia hematobium. Now later it was renamed as visceral blood fluid because it is found in the blood vessels, so it was named as visceral blood fluid. So now talking about its history, the Schistosoma hematobium were formerly restricted to the Africa only, and in the Africa, the schistosomiasis was reported from many years ago, from the millennium. That is, the eggs found in the eggs were found in the renal pelvis of an Egyptian mummy dating from 1250 to 1000 BC. So this schistosomiasis was common in Africa from ancient times. So also not only this, the schistosome antigen was identified by ELISA method in Egyptian mummies in 3100 BC. So this schistosomes is restricted in the Egypt part of the Africa. It was mostly common in the Egypt in Africa, and also it is now common in other parts of the continent. The adult worm was described by Bilharzia in Theodore Bilharzia in 1851 in Cairo. That's why the worm was formerly known as Bilharzia hematobium. And the life cycles of the worm was described by Leper in 1915 in Egypt. So this is the history of schistosome. Now Schistosoma hematobium. Now talking about the epidemiology of Schistosoma hematobium, the Schistosoma hematobium infects about 200 to 200 million people worldwide, and 200 million people worldwide. So this is the epidemiology. And talking about its distribution, the Schistosoma hematobium was prevalent through the Nile Valley, that is a part of the Egypt. So it was formerly prevalent in the Nile Valley, and it is also found in many other parts of the Africa and in West Asia. So West Asia, it was the Schistosoma hematobium was reported by Gurdgill and Shah in 1952 from India, from the Ratnagiri river in the India. So it was reported by Gurdgill and Shah in 1952. So it is also common in West Asia. Now, the morphology. Talking about the morphology of Schistosoma hematobium, it has two different morphological features. That is the adult worm and the egg. So, in the adult, the sexes are separate. That is, male and female are separate. So, now the habitat. The habitat, talking about the habitat of Schistosoma hematobium, it is found in the vesicle plexus and the pelvic venous plexus in the definitive host. So the male worm has a size of 10 to 15 millimeter by 1 millimeter and covered by a finely tuberculated cuticle. So male has a size of 10 to 15 millimeter and the breadth it is 1 millimeter broad. And the body of the male is covered by finely tuberculated cuticle. The male has two suckers in its body, that is the oral sucker and the ventral sucker. Here in the picture you can see there is the oral sucker, and this acetabulum is also known as ventral sucker. So this due to the presence of this ventral sucker, hematobium. So, Schistosoma hematobium has two morphological features, that is the adult form and the egg. And it is talking about its habitat. It is found in the pelvic venous plexus and the vesical plexus. That is in the veins surrounding the urinary bladder. And uh, the adult form are separate in their morphological features, that is the male and female are separate, and the male is broader than the female and the female is longer than the male, and the male has a size of 10 to 15 millimeter and it is 1 millimeter thick, and the body of male worm is covered by a finely tuberculated cuticle. And it bears two types of suckers, that is the oral sucker and the ventral sucker. Here in the picture you can see there is a oral sucker, that is the sucker surrounding the mouth is the oral sucker, and the ventral sucker or the acetabulum is found just beneath the esophagus. So, from the ventrals, just beneath the ventral sucker, there is the lateral borders of the male are rolled ventrally to form a gynecophoric canal in which the female worm is held. Here in the picture you can see there is a hollow cavity like canal which is called as gynecophoric canal, and in this gynecophoric canal the female worm is held in. So this male and female worm live in the close proximity. Though the sexes are separate, they live in close proximity. And the female worm has a size of 16 to 20 millimeter and a breadth of 0.25 millimeter. But the female in the male worm, whole body is covered by the tuberculated cuticle, but in the female, the cuticle is confined to the two ends, that is the anterior and the ventral end. Uh, and the gravid female or the mature female contains 20 to 30 eggs at one time within its uterus. So in the female there are 20 to 30 eggs within the uterus, and it may pass up to 300 eggs per day. The gravid female can pass up to 300 eggs per day. Now the other morphological form is the egg. Now talking about the egg is oval in its structure, and it has a brownish yellow colored cell, and the egg is non-operculated. So it does not float in the salt solution. So the shape of the egg is oval, and it has a brownish yellow colored cell with a terminal spine at its posterior end. The Schistosoma hematobium has a terminal spine at its one pole in the posterior side. The size of the egg is 140 micrometer long and 50 micrometer broad, and the egg consists of a fully developed mirazoidium within the egg. So the egg bear a fully developed embryo that is called miracidium. Due to the presence of this terminal spine in its posterior end of the egg, it can pierce the vesicle wall of the urinary bladder and pass into the lumen. So this terminal spine helps the egg of the Schistosoma hematobium to penetrate into the lumen of urinary bladder. The clinical features of the Schistosoma hematobium are mostly due to this egg, but not due to the adult worm. So we will talk about this in later. And the eggs are passed in urine more during the midday. Due to some unknown reasons, the eggs are passed in urine more during midday. During its life cycle, the eggs can also penetrate other sites of the definitive host, like the pulmonary aorta, and other sites, other ectopic sites during its life cycle. So the eggs, though they can pierce other sites, but they do not become sexually mature in these areas. But they can cause some tissue damage in those sites. So, like the eggs may also be found in the rectal biopsies. These eggs can be found in the rectal biopsy, but they are never passed in the feces. So now talking about the life cycles. The life cycle of the Schistosoma hematobium completes in two stages. That is, it is digenetic in its life cycle. So it requires two hosts for its completion of its life cycle. That is, the definitive host is the human, and intermediate host is the is the snail of the genus Bulinus. And the infective form is the Cercaria larva. So the transmission of this Schistosoma hematobium begins when human being comes in contact with the fresh water, which is contaminated with the urine of a infected person bearing the eggs of Schistosoma hematobium. So, okay, this is all for today. We will talk about the life cycle from in our next lecture. You have got another class, I guess. So we will talk about. We will start from the life cycle in our next lecture. Okay.